I'm Dr. Martin Handa of Asante Health, and today I'm welcoming Dr. Marco Ruggiero, a molecular biologist and medical doctor, to discuss with us his exciting development of an innovative immunotherapeutic tool named Immuno. Hi there.
Hi, thank you. Thank you so much for this beautiful introduction, and thank you for this great opportunity. I'm here at your service to try to explain this truly exciting development that occurred in the past couple of years or so.
Thank you. Thank you very much. It's a pleasure to have you here with us today as well. So we're looking forward to having our discussion today. So Dr. Ruggiero, first, please tell us about your training and work experience.
Well, unfortunately, this could be very long considering how old am I. I am 63, but I'll try to make it as short as possible. In 1980, I got my MD at the University of Florence. I have a specialization in diagnostic radiology at the University of Siena, Italy, and a Ph.D. in molecular biology at the Ministry of University in Rome, Department of University in Rome. In 82-83, I served in the Army as medical officer, and in 1984, I moved to the USA, working for Barrows Wellcome, Big Pharma, together with Dr. Eduardo Lapetina, and I had the honor to publish the paper in PNS, Proceedings of the National Academy of Sciences of the USA, sponsored by Sir John Vane, a Nobel laureate in 1982. And this paper was on protease inhibitors in the days when the AIDS epidemics just began to be recognized. In 1985, back at the University of Florence, Italy, I published a paper together with colleagues, Professor Vincenzo Chiarugi and Simonetta Vannucchi, on the supramolecular assembly of plasma heparin, the heparin that we have in our blood. And I mentioned this paper because this was the beginning of a development that eventually 33 years later led to the design and development of the supplement that will be the main topic of today's talk, Immuno. Then in 1987, I went back to the USA, this time working for the US government at the National Cancer Institute of the NIH in Bethesda, Maryland, working with Dr. Stu Aronson and Professor Peter Duesberg from Berkeley, California. I was working in those days on the genes responsible for cancer, the so-called oncogenes and signal transduction. In 1988, I had the opportunity to publish a paper in Science on signaling related to the EGF. Today, this gene is very well known for its involvement in breast cancer. And I began to be interested in what today is called immunotherapy. We didn't call it this way in those days. And in 1990, I published together with colleagues from the NCI and NIH, a paper on macrophage colony stimulating factor and human cancer. And please notice that this was about 4 years before Professor Yamamoto from Philadelphia published the first paper on the so-called GCMAF macrophage activating factor. I went back to Italy, I got tenure at the University of Florence, I became full professor of molecular biology. And in 2009, I began to be interested in what it is truly called now, and it was called then also immunotherapy. Immunotherapy, that is to harness the power of the immune system to fight diseases, mainly cancer and neurodegenerative diseases. And also I began to be interested in the role of the microbiome. Today, everybody is excited about the role of the microbes in our bodies, but in 2009, it was still something very little acknowledged that we have so many microbes in our body, we're only 10% human and so on. In 2010, I worked together with Dr. Yamamoto, and we published a study, we presented a study at the 18th International AIDS Conference in Vienna, Austria, demonstrating the effects of GCMF, this macrophage activating factor, in human monocytes that are human macrophages. In 2013, I began collaboration with Dr. Jeff Bradstreet, world authority in the field of autism. And in 2014, together with Bradstreet, we published a paper on transcranial ultrasonography as a tool for diagnosis and therapy in autism in a journal called Frontiers in Human Neuroscience. 1 year later, together with Bresrit again, just before his untimely death, we published in Frontiers in Neuroscience a paper where we hypothesized, we thought that we had found the pathogenesis of autism that is related to impaired flow of lymph from the brain through these deep cervical nodes. And this paper is now in the top 1% of all papers ever published by this journal. 2015, I moved to the USA, where I'm now. 2016, I began collaboration with another famous doctor in the field of alternative complementary medicine, Dr. Dietrich Klingert, originally from Germany, working in Seattle, Washington. And together with him, we developed a protocol that is called the Ruggiero-Klingert protocol, or RK protocol, published in the American Journal of Immunology, where we described a novel protocol for diagnosis and treatment of chronic conditions ranging from chronic Lyme, that is the expertise of Dr. Klingert, to autism and other chronic conditions. And in 2018, based on a number of papers and years of research in this field, I invented, so to speak, or I would say I designed and developed the Immuno, which is the supplement that will be discussed today. All in all, I have about 232 publications between peer-reviewed papers, book chapters, congresses, presentations, and so on. You can go in Google Scholar and you can find them all. You may find that I have about 3,500 citations as of yesterday. And some papers, like the one we wrote together with Bresrit, has a great number of views for a scientific paper, of course, 27,000 views. And as a matter of fact, as I said, it is in the top 1% of all of Frontiers articles. So this is a very short, of course, I will omit all the details of research, what I did, where. I think that these are the most important steps in my scientific career.
Fantastic. Thank you very much. And that brings us to the next question, actually. If you could please tell us about your current research interests, actually.
Well, you know, research is a matter of being curious, and I've always been very, very curious. When I was a student of medicine, I was fascinated by the topic of cancer. First of all, because in the mid-70s, we knew very little about cancer. We didn't know anything about genes and so on. And so it was truly a mysterious disease, but it was a disease that was different from all other diseases. So until then, medical researchers were concentrated on finding infectious agents. Cancer was something completely different. So this curiosity has never left me. And as of today, my main topic of research is still cancer, but at the light of today's achievements. So today, I would say immunotherapy of cancer as it relates to signal transduction. What does it mean? Cancer is a disease of communication. Cells don't understand each other, don't communicate well with each other, and they end up harming the host. So cancer cells, in a sense, are not enemies, are simply misguided, so to speak. And so the communication within the cancer cells, the communication between cancer cells and the cells of the rest of the body, and the communication inside the cells is truly fascinating, because the more we understand, the more we can do something, let's say, to fix the problem, to re-educate, without sending them to camps, of course, to re-educate the cancer cells so that they can go back to normal. This is something that is doable. It is called reversion of cancer phenotype. And as a matter of fact, there are a few papers that you can retrieve in PubMed demonstrating that you can revert the cancer phenotype. So you can make cancer cells become normal. One of these papers was published by a research group in 2015 in a prestigious cancer journal. It is called Anti-Cancer Research. And over there, we describe the negativization, as we say, of oncogene expression. So cancer cells don't appear cancerous any longer, also at the genetic level, through immunotherapy. And of course, today I'm focused on the study of immunotherapy as a way to harness the power of the immune system to fight cancer, but also other chronic conditions. As I said before, in 2009, I began to become interested in microbiome medicine, what today is called microbiome medicine, that is how the microbes influence our well-being and how are they involved in diseases, and how are they involved in the development and function of essentially all our organs and systems, including the immune system and the brain. Together with an author from the USA, Mr. Peter Greenlaw, we published a paper entitled Your Third Brain, indicating by third brain the information that is contained in the microbiome. I was the first to coin the words brain microbiome, because until recently it was thought that the brain was sterile. It is not. We have microbes in our brains, just as we have microbes in our gut, just as we have microbes on our skin. And those microbes inside our brains are as important as neurons and glial cells. They are essential for the functioning of our brain, of our mind, the way we think, the way we function. So the words brain microbiome as a sort of a primacy was published in a paper of mine that you can retrieve in PubMed in 2016. And today I'm working on studying the way microbial cells communicate within each other and with our cells. So still I'm sort of a researcher of communication, not communication between humans through Twitter or other media, but communication between cells, cancer cells and healthy cells, microbes and our cells, microbes with microbes, microbes with viruses, and so on. I am fascinated by this because, again, I think that as it often happens with humans, wars or fights are started because of lack of understanding, because of misunderstandings. And if this is true for humans, and it is true, it is even more true for cancer cells or microbes that out of a sudden become enemies of ours, whereas usually they're friends of ours, and so on. So communication between cells, you could say, is the main topic of today's research. In the field of autism, I'm still pursuing the role of ultrasonography that we started with Dr. Breastreet in 2013 in diagnosis and treatment, and of course, what is the role of the immune system and the microbiome in autism and neurodegenerative, neurodevelopmental diseases. And finally, this is sort of a hobby. I am working on something that may appear loosely related to whatever I've said thus far, that is gravitational time dilation and biological quantum entanglement. These are very exotic topics. You may think that gravitational time dilation, that is when you are closer to the center of gravity, time slows down. So you may think that these are things for physicists or for people interested in astronomy. Well, this is true, of course, but it is also of interest to the biologist and the medical doctor because we have seen that you can slow down the passing of time thanks to this idea, this hypothesis that is 100 years old by Einstein, of course, by simply introducing some gravitational attractor in the proximity of DNA. So small things, they work like big planets and stars and black holes. And biological quantum entanglement also is something very fascinating that also influences the way this supplement immuno works. So these are my main topics of interest, my main topics of research in these days, and I shall be more than happy to elaborate on these if these are of any interest to you.
Absolutely, fascinating. And I guess that also leads us next to what led you to develop immuno.
Oh, well, this is something that I would say came natural. Came natural by working in the field of immunotherapy, and came natural by working together with Dr. Yamamoto, who was the one who in 1994 first coined these words, GCMAF, macrophage activating factor, and had the idea that this way of stimulating cells of the immune system, macrophages, could lead to immunotherapy of cancer and HIV infection. We began working with him, and as I said, in Vienna, we presented our first results at the most prestigious World International AIDS Conference. And then we kept on working on GCMF back at the University of Florence. We published in a journal that is devoted to cancer immunology and immunotherapy, and it is called Cancer Immunology Immunotherapy, where we demonstrated that this factor not only activated macrophages, but also inhibited what is called angiogenesis. Angiogenesis is that process that leads to the formation of new blood vessels. You know, when cancer grows, it needs blood and oxygen, but blood vessels aren't there, so the cancer has to build its own blood vessels, like lines of supply. And this process, of course, leads to greater cancer expansion. So if you could block this process, you deprive the cancer of its blood, its oxygen, its nutrients, and so you can stop the growth of cancer. We demonstrated that immunotherapy also worked in this sense. In 2013, still working on this factor, we were the first to propose a molecular model on how this factor interacted with cells. And then we began to realize that maybe the protein of the GCMF was not the most important part of the molecule, but the fatty acids and the vitamin D3 that were bound in a multimolecular complex were responsible for the biological effects that we and many others were observing with this factor. And this leads back to the original idea of Yamamoto, who in 1998 thought that GCMF could work as an adjuvant. Adjuvants are those oil-water emulsions that are used in vaccines to enhance, to improve the immune response to a given vaccine. So this was Yamamoto's idea of 1998, and we began to realize that this hydrophobic, hydrophilic, that is a sort of oil-water interaction at the cellular level, was responsible for the effects that we were observing. At about the same time, so between 2013, 2014, several papers came out because of the interest induced by this type of novel immunotherapy. So many papers confirmed what we and others had observed, but also many papers they began to question the interpretation of the results. And for example, there was a paper demonstrating that people with cancer, they have normal levels of GCMF. So if you have a normal level of something and still you have cancer, quite evidently there is no deficiency of that factor, and quite evidently, even if you gave that factor and you observed some effects that weren't due to some deficiency. And so these and many other inconsistencies at the molecular and biochemical level that are too boring to be mentioned today led us to rethink, or let's say reinterpret, the results that we and many others had observed. Because again, the effects of GCMF in cancer have been independently replicated by scores of researchers, and also the effects of GCMF and autism have been replicated. So it worked, this type of immunotherapy worked, but maybe the original interpretation that everything was due to a given protein that is called GC protein was not correct. So in 2016, we came up with the idea that we published in a journal called Medical Hypothesis, that is devoted to publish exactly hypothesis, that the active site of GCMF was indeed not a protein, but a glycosaminoglycan that is called chondroitin sulfate. That is complexed with hydrophobic, sort of oily molecules such as oleic acid and vitamin D3. And retrospectively, this was consistent with our paper published in Anti-Cancer Research in 2014, where that is actually, and very aptly I would say today, entitled Oleic Acid, Deglycosylated Vitamin D Binding Protein, Nitric Oxide, and Molecular Triad Made Lethal to Cancer. So we didn't know at that time, but we had already focused on the hydrophobic, hydrophilic, let's say oil-water emulsion as the responsible for the clinical and biological effects. So this intuition led to the development of a supplement called Rerum that was an emulsion of chondroitin sulfate, oleic acid, and vitamin D3. But then from Rerum, where we observed excellent results, we moved to this current development that is called immuno, that represents a sort of a quantum leap, a big leap, because it features non-animal derived ultra-pure chondroitin sulfate. So we're not dealing with human blood derivatives. We are not dealing with animal-derived compounds at variance with GCMF or Rerum, and ultra-pure phosphatidylcholine and D3. So we're not dealing with mixtures of oils because we found which one of the several, say, oily molecules were responsible, and D3. So immuno is a sort of the culmination of more than 30 years of research. You may remember that in 1985, when I was a young postdoc at the University of Florence, we published this paper demonstrating how glycosaminoglycans are assembled in human blood. And it is from that inspiration of more than 30 years ago that through a series of successive and very logical steps, I designed and developed immuno. As of today, we have 3 peer-reviewed papers published on immuno, and a 4th one has just been submitted describing the effects of immuno in autism and how it is integrated in the Ruggiero-Klinger protocol, and we hope that soon this paper will be accepted and published. So it's a logical sequence of events that led to immuno, did not come out of the blue, and it is based on more than 30 years of research.
Absolutely brilliant. And I guess the next thing I'd be wondering is, please describe the many functions of immuno. So.
Well, immuno, again, is a supplement, but it is unique among supplements for a variety of reasons. First of all, its molecular structure. As I said, oil-water emulsions, or in scientific jargon, hydrophilic, hydrophobic emulsions, have a number of effects. Immuno is modeled upon the early protocells, the first forms of proto-life that appeared on Earth about 3.5 billion years ago. So it is sort of a self-assembly where the core is this low molecular weight, non-animal derived chondroitin sulfate, a glycosaminoglycan. And this ultra-pure, homogeneous, low molecular weight chondroitin sulfate is inherently nano-sized. It comes in nanometers. So you don't need to nanosize it, it is already nano-sized. Now, around this core, you have a bilayer, that is 2 layers of ultra-pure phosphatidylcholine. Now, this molecule, they self-assemble. So you don't need to tell them how to interact with each other because they do this automatically. And this type of self-assembly is what is thought today and for many years, to be at the origin of life, following a principle that is called negentropy. Negentropy means negative entropy. It was Erwin Schrödinger, a physicist, who in 1944, during World War II, wrote a book, What is Life? And he first talked about this self-assembly of molecules in the context of negative entropy. And vitamin D3 is intercalated in this structure. So, first of all, the molecular structure is unique. Secondly, because of the small size of its components, low molecular weight, chondroitin sulfate, and of course, phosphatidylcholine, that is a single phospholipid, it spontaneously forms nanoliposomes. So I'm sure you're familiar with the concept of liposomes that are excellent delivery systems to deliver a number of supplements and drugs. Take, for example, vitamin C as an example. These liposomes, self-assembled liposomes, are in the nanometer range, so are nanoliposomes. And so you have nanoliposomes without the need of doing anything. So you don't need to use ultrasounds as you usually do when you want to make liposomes for vitamin C, for example, or other technicalities. We could say again that it is the most primeval form of biological organization. And since all biological reactions depend on a surface phenomena, and the molecules, they react and recognize each other at the level of their surfaces. Surface biochemistry is essential in all biochemical processes. So because of this, and because you are working at the level of nanometers, you have an enormous efficiency, because just a little quantity of immuno exerts great effects at the biological level, because it is nano-sized and it is nanoliposomes. And again, it is a spontaneously nano-sized. You don't need to do anything. It's a self-assembly. Now, it is important that not only it is nano-sized, but it is also liposomal. So you have a sort of a nanoliposomes. Why it is important? Because, for example, when we talk about vitamin D3, vitamin D3 has a huge number of effects, but also it has some side effects, like, for example, hypercalcemia. So if you take too much vitamin D3, you end up with too much calcium in your blood, with a number of unpleasant consequences. But it is known that if you put vitamin D3 into liposomes, you significantly reduce the hypercalcemia. So in our case, everything is done without us telling the molecules what to do. Because in our case, in the case of immuno, vitamin D3, it is naturally inside liposomes that are in nanometer range. And because of these peculiarities, it works on different levels. First of all, it works because its components, chondroitin sulfate, phosphatidylcholine, and vitamin D3, they have known biological effects, known healthy effects. They have been on the market as supplements for decades, and everybody knows that vitamin D3 or chondroitin sulfate or phosphatidylcholine have a number of effects. What is the novelty? Where is the difference? In immuno, because they are organized in such a unique molecular structure, their effects are amplified. And also because they are nano-sized and they are organized as liposomes. So this favors bioavailability and cellular and molecular interaction. So you need a very low dose. Now, what are the known activities of each one of the components? Very many. I'll try to summarize. Chondroitin sulfate has a number of known effects. For example, it is known that it has anti-inflammatory properties. As a matter of fact, it is used in the context of rheumatoid arthritis. And it has effects on wound healing, and it has immunostimulating and anti-cancer properties that are known since the 60s of the last century. It was Dr. Freund from New York Hospital who described these immunostimulating or immuno-augmenting, as he called them, activities and anti-cancer effects of a very primitive bovine cartilage extract. It also has neuroprotective effects. That is, it is very useful in neurodevelopmental and neurodegenerative conditions. What about phosphatidylcholine? Reduces inflammation, enhances the cognitive function, shows positive effects on mood, daily functioning, cognition of elderly patients with Alzheimer disease or dementia. So it has a number of effects that again are amplified by its molecular assembly in immuno. And vitamin D3, you know, it's a very complex hormone. It is much more than a vitamin. It's a hormone that is involved not only in calcium homeostasis, but also in the integrity of the immune system. Vitamin D deficiency, as you know, is linked not only to rickets, but also to inflammatory and long latency diseases such as multiple sclerosis, rheumatoid arthritis, again, inflammatory conditions, tuberculosis, diabetes, and various types of cancer. Now, up to the point that in 2008, authors from the Medical University of South Carolina wrote a paper entitled, "Does vitamin D make the world go round?" So it was a kind of a rhetorical question, and just to show how important is vitamin D in the well-being, in the homeostasis of our body. Then there is, in addition to these, I would say, mainstream or classical effects, there is the epigenetic level. Now, because we have nanoliposomes containing low molecular weight, chondroitin sulfate, and vitamin D3, now you have molecules that can penetrate inside the cells. So because of this configuration, the low molecular weight chondroitin sulfate and the vitamin D3 can enter into the cells and can interact with DNA. Now, this becomes complicated, and I hope that it is not too boring, but the interaction of chondroitin sulfate with the DNA leads to results similar, if not identical, to those of a class of drugs that are called histone deacetylase inhibitors. Now, these are compounds that have proven useful in cancer. For example, one of these is called vorinostat, and it's very useful in a number of different cancers, in psychiatric and neurological diseases, in chronic inflammatory diseases, and so on. So essentially, immuno, by penetrating into the cells and by having the chondroitin sulfate bound to DNA, exerts effects comparable to those of histone deacetylase inhibitors that again are very, very effective drugs. Now, because of this, this interaction with DNA normalizes gene expression. Now, this leads to some slippery slope because normalizing gene expression leads to the concept of adaptogens. I don't know if you're familiar with adaptogens, but it is a term that has had a lot of attention. Now, the concept of adaptogens was originally created in 1947, so many years ago, to describe a substance, originally some herbal preparations, botanicals, that may increase resistance to stress. And adaptogenesis was later applied in the former Soviet Union to describe remedies, natural remedies, thought to increase the resistance of organisms to biological stress by essentially rebalancing the homeostasis of organisms. Another definition for an herb to be considered an adaptogen requires that it must be non-toxic, non-specific, and it has, of course, to affect the physiology of the organism, and so immune response to all these requirements. Problem is that most of the studies conducted on adaptogens were performed in the former Soviet Union, Korea, and China before the 1980s. And in the United States, the Food and Drug Administration issued a warning recently in 2013 discouraging the use of this word because they say there is not sufficient scientific information behind. For example, there was a company based in Washington and they advertised something as an adaptogen, and the FDA said, no, you can do this, whatever the case. So whether you want to use the word adaptogen or not, there is no doubt that by regulating gene expression, as immuno does, as histone deacetylase inhibitors like vorinostat do, you definitely rebalance the expression of genes, so you rebalance the working of DNA. Another mechanism of action is the role of as an adjuvant. I don't know whether I'm pronouncing this known correctly or not. It comes from the Latin adjuvare, which means to help. Now, adjuvants are molecules that are designed to help boost the immune response, in particular when you are given a vaccine. And in 1998, Yamamoto thought exactly that GCMF was working as an adjuvant. But there is something a little bit curious or mysterious. Now, as you know, vaccines are given in billions of doses to humans and animals with undisputable efficacy. So for the moment, we don't enter into the controversy about vaccines at a certain age in humans. But vaccination works, on this there is no doubt. But vaccination works because of adjuvants. And the mechanism of action of adjuvants in vaccination remains what a recent paper called immunologist's little dirty secret. Because essentially, nobody knows very, very well why you need an adjuvant, which is a rather brute substance like a water oil emulsion, to boost an immune response. Shouldn't the antigen be enough to teach the cells of the immune system to fight against a virus or a bacteria or something else? No, they are not. Because in order to activate a T and B lymphocytes, so to have effective immunization, if you do without adjuvants, no matter how ultra pure the antigen is, no matter how ultra pure the vaccine is, you do not achieve effective immunization. So you need these things. And immuno was designed to be sort of an universal adjuvant. So it helps to boost the immune response by itself, but also it helps to boost immune responses when you want to go another route. For example, and maybe we can talk about this later, for example, when you want to employ vaccines against cancer or vaccines against Alzheimer's disease, so the so-called therapeutic vaccines. In that case, something like immuno might definitely boost the immune response. And then there are two other mechanisms of action that are a little bit more exotic. The so-called non-chemical DNA signaling. Until now, we have talked about immuno, considering it just like a kind of a peculiar, kind of innovative, but still a molecule that interacts with other molecules. So in your mind, you can think that chondroitin sulfate goes and binds to DNA, vitamin D3 goes and binds to its receptor, and then binds to DNA, and so on. So classical biochemistry, classical chemistry, biochemistry, recognition between two different surfaces, and so on. But there is something more. Think that chondroitin sulfate is a highly charged molecule. So it has a very dense concentration of negatively charged groups because of the sulfate groups and the carboxylic acid. So think of it like the negative pole of a battery. So a number of negative charges on the surface of chondroitin sulfate. Phosphatidylcholine, on the contrary, has positive charges on its head, the choline. So you have positive charges, so the other pole of a battery. So here you have a complex of positively and negatively charged molecules put together, just as if you have a battery with two poles, negative and positive. Point is that chondroitin sulfate and phosphatidylcholine assembled in these protocellular structures that we first observed in 1985, as I said before. Now, they are in our bloodstream, and they are propelled at speeds varying from 40 centimeters per second in the aorta, just after the heart has pumped the blood, down to 0.03 centimeter per second in capillaries. So they have a positive and negative acceleration. So they accelerate first and they brake then. And every time that an electrical charge is accelerated, whether positively or negatively, so whether it accelerates or decelerates, there is a generation of an electromagnetic field. Now, electromagnetic field of these types, of course, are variable because the speed varies, and are individual because the speed of your blood is different from mine, whether you are exercising or not, whether you have atherosclerotic plaques or not, whether your arteries are tortuous or are straight, whatever. So it's very, very individual. So this radio signal that is associated, because it's nothing else, a varying electromagnetic field is a radio signal that is generated by the molecules of immuno every time they're pumped out from the heart. These radio signals, they interact with DNA. Now you may think, how can they interact with DNA? Is DNA an antenna that is able to receive radio signals just like your radio? And the answer is yes, absolutely so. And actually, Dr. Stefano Atterini and myself, in the Encyclopedia on Cancer and in many other papers, we described how DNA works like a fractal antenna. So it is just like the antenna. It's a very, very long molecule. So it's a very long molecule, very long antenna. We have about 2 meters of DNA in each cell of our body. So think how long it is and how many kilometers or miles of DNA we have in our bodies. So DNA works as a fractal antenna, able to send and receive signals under the form of radio waves. Now, the radio waves, or varying or variable electromagnetic fields generated by immuno in our blood, and because they are reminiscent of the organization of protocells in our body, they are supposed to reset the functioning of DNA. Just like you reset your computer when you have downloaded too many viruses or you have made too many mistakes, so you're able to reset your computer. We think that these radio signals generated by the flow of immune in our blood is able to reset the functioning of DNA. Now, this is very important when DNA doesn't produce the right communication, like in cancer, for example, or in autism, or in neurodegenerative diseases. That's where my curiosity about communication in cells came to help. So cells don't communicate uniquely through hormones or neurotransmitters or molecules. They also communicate with each other through radio signals. And we think that immuno is able to restore or to reset the original radio signal. And then the last one is the so-called level of the biological quantum entanglement, what Einstein's described, who never accepted so much this phenomenon, described it as a spooky action at a distance. Now, we know today that the quantum entanglement, of course, is not only limited to the realm of physics, but it is very well present in biological processes. So we know that it is involved, and probably it is at the basis of human consciousness, thanks to the work of Professor Stu Ameroff at the University of Arizona in Tucson, not too far from here. We often see each other, and it's always enriching to speak with him. So we know biological quantum entanglement in human consciousness. Bird navigation, birds could not find their ways in the sky if not for biological quantum entanglement. Photochemical reactions like photosynthesis and gene expression, that is what it is of our interest. Now, all living beings somehow are entangled because we all derive from the primitive ancestors whose appearance on Earth as primitive, primeval protocells dates back to about 3.5, 3.8 billion years ago. So we are all entangled. This concept has been widely described not only by biologists, but also by philosophers like Deleuze and Guattari, French philosophers who in the 70s proposed this idea of a rhizome that is sort of a root that encompasses all forms of living beings of today and yesterday and probably of the future on Earth. And I published a paper how this concept relates to immortality in 2017. So immuno, by resembling the primitive protocells, is able to send a signal through quantum entanglement that again resets or brings to equilibrium the functioning of not only DNA, but probably other structures as well. I know I've been rambling around complex concepts, so please feel free to ask for any clarification you may need.
No, that is fantastic. The next thing was, how can immuno be administered, and what is the benefit of the different routes?
Well, there are different routes. First of all, let's remember that we have to follow rules. So first of all, you have to find out in your country or in your state, if you are in the United States, for example, which are the rules for administration of supplements and what is legal, what is not. And of course, we encourage everybody to stay on the good side of the law. So whether you are a naturopath or a medical doctor, follow the rules, don't administer things unless they are authorized in your state or your country. This having been said, and I think it's quite logical, we have to consider that immuno is manufactured as a sterile, natural nanoemulsion. And these three adjectives, sterile, natural, and nanoemulsion, provide a great deal of versatility as far as bioavailability and therefore ways of administration are concerned. Now, the most common way of administering a supplement is through the oral route, and it definitely works. But also, since we're talking about the mouth, we can talk about sublingual administration. Under our tongue, the mucosa is very thin, highly vascularized, and we know for decades that things that are put under our tongue are promptly absorbed, they go into the bloodstream and they exert the effects. And immuno, because it is a nano-emulsion, is perfectly fit for this way of administration. And another way of administration, if you want to target, for example, the immune system in our lungs, because maybe there is some pathology where you wanted to target the immune system in the lungs or in the bronchi, is a nebulization with a common nebulizer. Again, since it is a nano-emulsion, no problem in being nebulized. Or you may wish to use a nasal spray to target the brain through the olfactory nerve. Now, the olfactory nerve is the trade union between the nose and the brain, inside the brain, so you don't need to drill a hole in the skull to reach the brain. You simply have to use some deep nasal spray, and the olfactory nerve will serve like a door, so these molecules can climb, so to speak, into the brain. And again, since they are oily emulsions, it's a water oil emulsion from the physical chemical point of view, they can reach the brain through the olfactory nerve. And then, of course, there are the injections. Subcutaneous injections are the best way to deliver anything if you want to stimulate the immune system. As a matter of fact, when you want to vaccinate somebody, you use most of the times the subcutaneous or intradermal injection, because this is the best way to stimulate or rebalance the immune system. And when you have an adjuvant like immuno is, you have the maximum immunization response. Now, there is something interesting. Intradermal vaccines or subcutaneous vaccines offer the advantage they could also be self-administered, and there are proposals referring to vaccines against flu, for example, to send them through the mail so that people can inject themselves, by so doing, saving a lot of money, and according to the proponents of this idea, by improving the health of populations that are in areas of the world where you don't have too many doctors or are difficult to reach. So this feature of being a sterile nanoemulsion allows a great versatility. Now, there is also another way that is gaining momentum in these days, that is the transdermal delivery. Transdermal delivery means through the skin, possibly when it is combined with a cream or a lotion. We worked on transdermal delivery systems for glycosaminoglycans like chondroitin sulfate. Actually, we were working on heparin, that is a molecule closely related to chondroitin sulfate, since 2006, so now it's 13 years. And we were delivering heparin through the skin and observing the biological effects of heparin once it was delivered through the skin. I think you know that when heparin is given as an antithrombotic drug, you have to inject usually in the belly, and you end up with bruises, not so nice. And then, of course, injections, they have a number of side effects. So because of this, we wanted to find a way to deliver heparin through the skin without resorting to injections. And we succeeded. We published this paper in 2006. And of course, now we use that expertise to deliver immuno, which is made of chondroitin sulfate that chemically is extremely similar to heparin. And since, again, it is a nanoemulsion, it's very easy for the immuno to cross the skin barrier. And beyond the skin barrier or beneath the skin barrier, recently it has been described that there is a new organ. It is called interstitium. It is a complex network of microscopic tubules that connects all organs and systems. And most important, it connects the immune system to all organs. So I anticipate that since immune is very young, that this will be a main way of administration. And the transdermal delivery of immuno will constitute a major breakthrough in delivery systems, and it will have a number of potential applications, even more than what we are thinking today.
Fantastic. That's very interesting. And I guess the next question would be, what conditions are immuno likely to be a benefit for?
Well, first of all, again, let's remember that immune is a supplement, and let's remember that as medical doctors, we are bound to follow the standard of care that, of course, is different in every different country or state, and I strongly recommend all therapists and doctors to follow the standard of care for any individual disease. But this having been said, in the context of complementary, not alternative, natural immunotherapy, like, for example, nutritional interventions, immuno may have a number of indications. Essentially, all the conditions where chronic inflammation is present, where imbalances of the immune system and neuroprotection is needed. And this covers essentially most chronic conditions, including, of course, cancer. For example, autism, neurodevelopmental disease. There is neuroinflammation in the brain, and this has been amply demonstrated. The glial cells are activated, a number of markers of neuroinflammation are present. And also there are imbalances of the immune system, and of course neuroprotection is needed. So autism could be a prime example of a condition where immuno could prove useful. And as a matter of fact, just a few weeks ago, Antonucci, a renowned world expert in autism, together with us, published a paper describing clinical cases treated with immuno. And many more cases will be presented at a conference in Romania in the next few days, and then at the World Conference on Autism in Chicago at the end of May, called Autism One. Let me quote just 3 of these cases very, very shortly. In one case, the behavioral symptoms are described by a score that is called ATEC, Autism Treatment Evaluation Checklist. After 2 months of treatment with immuno: bedwetting, wet pants diapers, soiled pants diapers, diarrhea, constipation, eats too much, too little, not sensitive to pain. Now, all these symptoms of autism, they improved from moderate to not a problem. So essentially, they were no longer there. And this indicated a complete normalization of these very significant symptoms of autism in this subject. Another subject: shows no affection, fails to greet parents, avoids contact with others, dislikes being held, cuddled. All these symptoms improved from very descriptive, so they were there, to not descriptive. So again, weren't there anymore. Complete normalization of these symptoms. Another subject: symptoms described that the queries: does not imitate, disagreeable, not compliant, indifferent to being liked. Now, the answers showed improvement from somewhat descriptive to not descriptive. Again, normalization of behaviors. And Antonucci, I know, he has many more cases. These are the cases that were published. So essentially, we are observing these effects of immuno in autism that are due to these, to the complexities of mechanism of action that I quoted before. Probably most important are the effects on neuroprotection, anti-inflammatory effects on the brain, but also restoring the immune system at the level of the deep cervical lymph nodes, the lymph nodes we have here, where the flow of lymph coming from the brain passes through. So, and this goes back to the paper together with Bradstreet about the pathogenesis of autism. And talking about other conditions that might benefit from this complementary approach complementary approach based on immuno, I can tell you that in Germany, there is a major hospital that is recruiting patients to be treated with immuno in the context of a complementary immunotherapy. Now, the first reports, they have not completed the study, but the first reports are extremely encouraging. And I've been told that traditional oncologists are amazed at what they're observing. Essentially, we are talking about late stage cancer patients where the traditional oncologists had lost all hopes, and against all anticipations, against all hopes, these patients are doing very, very, very well. And I don't anticipate the actual results because this will be the object of scientific papers, but let me tell you that the results that we're observing are very encouraging. But then, as I said before, it can be, immuno can be very interesting as an adjuvant in the context of therapeutic vaccines. Let me spend a word, because usually in the field of alternative or complementary medicine, everybody seems to be against vaccines, and vaccines are linked to all types of side effects. There are anti-vax movements and so on. I don't want to enter into this field at this moment, but I want to say that the vaccines I'm referring to right now are not the conventional vaccines like measles, rubella, and mumps, but I'm talking about therapeutic vaccines. So those vaccines that are being developed to cure chronic conditions like cancer, Alzheimer's disease, multiple sclerosis, or HIV infection. I think it was only yesterday that in the prestigious journal Nature Medicine, researchers working at New York's Mount Sinai Hospital published a paper entitled Systemic Clinical Tumor Regression with In situ Vaccination. So essentially what they observed in cancer patients, late stage cancer patients, they vaccinated these patients with some antigens that sort of educated the immune system to fight the cancer cells, and there was a tumor regression. So the tumor shrunk because of this vaccination. So these are the vaccines I'm talking about. In these types of vaccines, immuno could provide a formidable boost to the immune system, so it would amplify, would enhance the effectiveness of these vaccines. And as a matter of fact, in 2017, I published a paper entitled A Novel Method to Enhance Immune Responses Induced by HIV DNA Vaccinations, where I described the mechanism of action of immuno as far as enhancing immune responses to all types of therapeutic vaccines. So this paper was born out of the observation that Russian researchers, very recently, not from the old times of the Soviet Union, very recently they published a paper describing a HIV vaccine, HIV DNA vaccine, that was effective in 100% of the participants, but the immune response was weak. So that's where the idea of using immuno came from. It is, okay, they found a good vaccine, but the immune response that they are able to induce is weak. So why don't we simply potentiate this immune response by using something like immuno? And so this is where the paper came from. So I think that there are many conditions that can benefit from the use of immuno. Not only those, let's say, traditional for immunotherapy, but also some innovative conditions where all these properties of immuno may prove truly useful.
Wonderful, wonderful. I mean, first off, that's really helped in clarifying a lot about what immuno is about. And next off is just congratulations on such an innovative immunotherapeutic tool. So thank you, Dr. Ruggiero, for meeting with us today and sharing this exciting information on immuno. We feel sure it will be of great benefit to many people in the coming years, and thank you a lot for meeting with us and having a chat.
Thank you for this great opportunity. Thank you so much.
Thank you.
Bye-bye then.